Hello everyone, welcome to my YouTube channel. For today's video, we will discussing the second part of cell. Let's see. Let's start with the mitochondria. Mitochondria is a double membrane bound. We can see the diagram of mitochondria. First is outer membrane, then there is inner membrane. Between outer and inner membrane, there is intermembranous space. Here's ribosome is of 70S. And here mitochondrial DNA. Here DNA is circular DNA. In mitochondria, there is circular DNA. And we can say mitochondria is a semi-autonomous cell organelle. Next, this mitochondria is absent in mammalian RBC and prokaryotes. Under the microscope, these mitochondria are easily visible when it is stained. And stained by which dye? By Janus green. This question has been asked, mark as important. Number, shape and size of mitochondria per cell are variable depending on physiological activity. Depending on the physiological activity, mitochondria का number, shape और size vary करता है. कैसे shape होते हैं? Sausage shaped or cylindrical, having a diameter of 0.2 to 1.0 micrometer and length 1.0 to 4.1 micrometer. See, we have seen that mitochondria is a double membrane bound structure with outer membrane and inner membrane, which divides into two membrane into aqueous compartment. And this compartment, outer compartment and inner compartment, it is called matrix. Inner compartment में क्या होता है? Matrix. Next, inner membrane forms a number of enfoldings called cisternae towards the matrix, which gives the surface area, which increase the surface area. And these two membranes have their own specific enzymes associated with the mitochondrial function. And then this matrix possesses a circular DNA with few RNA molecules, ribosomes, and components for protein synthesis. And this mitochondria divides by region. क्या काम करता है mitochondria? It is the site of aerobic respiration or Krebs cycle. This Krebs cycle we will discuss in our further video when we will discuss respiration in plants. And they produce energy in the form of ATP. That's why we call mitochondria as a powerhouse of cell. Next is plastid. This plastid is also double membrane bound organelle. This plastid is also double membrane bound, and this plastid is found only in plant cells. Double membrane and only present in or found in plant cell only. Okay. These plastids are found in plant cell and in eukaryotes. Large size, which is easily observable under the microscope. Means here there is no need of any stain to see the plastid under the microscope. Plastid contains pigments. First is leucoplast. This leucoplast is a colorless plastid of varied shape and size. तो क्या स्टोर करता है? न्यूट्रिएंट्स. कलरलेस प्लास्टिड है और स्टोर करता है न्यूट्रिएंट्स. और ल्यूकोप्लास्ट में इंक्लूड होते हैं 3 टाइप्स: अमाइलोप्लास्ट, इलायोप्लास्ट एंड एलुरोप्लास्ट. एलुरोप्लास्ट कंटेन करता है, क्या स्टोर करता है? प्रोटीन. अमाइलोप्लास्ट स्टार्च और इलायोप्लास्ट ऑयल एंड फैट्स. नेक्स्ट इज क्रोमोप्लास्ट, विच कंटेन फैट soluble carotenoid pigments like carotene, xanthophyll, which gives yellow, orange or red color. Which gives yellow, orange or red color. This carotid we get red color, and this xanthophyll we get yellow color. Next is chloroplast, which contains chlorophyll and carotenoid pigment. Next is chloroplast. Chloroplast is also a double membrane bound organelle, which is mainly found in mesophyll cells of the leaves. This chloroplast is a lens-shaped, oval, spherical, discoid or ribbon-like organelle. The chloroplast length is 5 to 10 micrometer and width is 2 to 4 micrometer. This chloroplast, we can say it is bigger than mitochondria. This chloroplast we can say bigger than mitochondria. The inner membrane of chloroplast is less permeable, and the space limited by the inner membrane of the chloroplast is called stroma. जैसे inner membrane के अंदर जो space है chloroplast के उसको हम stroma बोलेंगे, and it contains many organized flattened membranous tracts like called thylakoids. See the diagram. This is inner membrane, we can see outer membrane, and between outer and inner there is intramembranous space. Okay. Now this inside we can see this fluid. We can see this inside fluid is called stroma, and this disc-like structure is thylakoid, and this group of disc called granum. And this granum is connected by stroma lamina. This is connected by stroma lamina. See here with a rough diagram. This is outer membrane. Inside there is inner membrane. This space called stroma. A group of disc-like structure. This called granum. Single disc called thylakoid. And this group is connected by lamella. Okay? Okay? Next. Membrane of thylakoid encloses a space called lumen. Chlorophyll pigments are present in the thylakoid, which means the light reaction. This thylakoid is site of light reaction. Next. Thylakoids are arranged in tracts called grana or the integranous thylakoid. There are flat membranous tubules called stroma lamella which connecting the thylakoids of the different grana. We have seen. This stroma contains small double-stranded circular DNA, ribosome, enzymes for synthesis of carbohydrates and protein. Inside the stroma, it contains small double-stranded circular DNA, ribosomes, and enzymes. And what they are doing? They are synthesizing the carbohydrates and protein. These chloroplast ribosomes are smaller, 70S, than the cytoplasmic ribosome, 80S. Next is ribosome. This ribosome is also a non-membranous granular structure. Who composed this ribosome? George Palade. In 1953. And this ribosome are composed of ribonucleic acid, RNA, and protein. It is first observed by George Palade, 1953. This ribosome is the site of protein synthesis, so that's why we call ribosome as a protein factory. Protein synthesis ribosome कहता है, इसीलिए हम उसको protein factory भी बोलते हैं. In eukaryotic ribosome, we can say there is subunit that is 60S and 40S, and whereas prokaryotic ribosome there is 70S. And in eukaryotic together form 80S. Next, cytoskeleton. This cytoskeleton is a filamentous proteinaceous structure which is present in the cytoplasm, and it consists of microtubules, microfilaments, intermediate filaments, and क्या करता है ये मैकेनिकल सपोर्ट देता है, मोटिलिटी देता है, and also maintain the shape of the cell. Microtubulin ट्यूबुलिन से बनता है, और माइक्रोफिलामेंट एक्टिन से बनता है, और जो इंटरमीडिएट फिलामेंट है ये इन दोनों के बीच का होता है. नाउ मूव टू नेक्स्ट इज सिलिया एंड फ्लैजेला. क्या काम होता है सिलिया और फ्लैजेला का? मूवमेंट में हेल्प होती है इनसे. दिस इज सिंगल membrane bound organelle. They are hair-like outwards of the cell membrane. Cilia, the small structures which work like oars. Oars means chaku. नाव में जो हम यूज करते हैं ना आगे बढ़ने के लिए, वो विच कॉजेज द मूवमेंट ऑफ द सेल और सराउंडिंग फ्लूइड. फ्लैजिला एज कम्पेयर टू cilia, flagella is longer, responsible for self movement, and we have studied that prokaryotic and eukaryotic in both flagella are structurally different. Cilium and flagellum are covered with plasma membrane, and their core axoneme has many microtubules running parallel to the long axis. And this axoneme has 9 doublets of radially arranged peripheral microtubules, which is a pair of central microtubules, which is called 9 plus 2 array. See here, we can, if we talk that in center there are two. In center there are 2 centromers, and in radially there are 9 doublets. 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, which means this 9, this plus 2, 9 plus 2. This in the center there are 2 center microtubules, and this peripheral microtubule doublet, which is 9, and they both are connected with inter-doublet bridge. And if we talk about this radial spoke are also 9. Okay. The central tubules are connected by bridge and are enclosed by central sheath. Connected to one of the tubule of each peripheral doublet by a radial spoke. There are 9 radial spokes, and these are interconnected by linkers. And this cilium and flagellum emerge from centriole-like structure called the basal body. This is important for the exam point of view. Next is centrosome and centriole. Centrosome and centriole. This centrosome and centriole is non-membrane bound. These are non-membrane bound. This centrosome is an organelle which containing 2 non-membrane bound cylindrical structure called centrioles, and they are surrounded by pericentriole material. And this centriole lie perpendicular to each other, which make up of 9 evenly spaced peripheral fibrils of tubulin. See, this is centriole. Here it spoke, one spoke, radial spoke, and here is the triplet. Like this, there are 9. And each of having a triplet. Centriole lie perpendicular to each other, which is made up of 9 evenly spoked fibrils of tubulin, and this peripheral fibril is a triplet, and the central part of the proximal region of centriole is also proteinous, which is called the hub, and it is interconnected with tubules by radial spoke, and this radial spoke is also made up of protein. And the basal body of this centriole, from this centriole the basal body arise a spindle apparatus when during cell division. Cell division के time पर they give rise to spindle apparatus. Okay. Next is nucleus. We have studied that nucleus was discovered by Robert Brown in 1831. This material of nucleus was stained by the basic dye. We have studied that basic dye. What is that? Acidocarmine. This is NCERT example question. And this nucleus is a double membrane bound structure. Normally, a cell has only one nucleus, and some cells have more than one. Mature RBC and sleeve tube cells has lack of nucleus. और सारे function of cell surrounded by nucleus. You can see here the diagram. Nuclear membrane, nuclear pore. This nuclear pore is for exchange or the supply of protein and carbohydrates. Nucleolus. Let's see. Nuclear envelope. This nuclear envelope is a double layer membrane, which is a space between, which is called perinuclear space, and it is a barrier between material present in nucleus and cytoplasm. इन दोनों को mix up नहीं होने देता है, and which that outer membrane usually remains continuous with ER and also be a ribosome on it. We have seen that ER is covered outer membrane of nucleus, A nuclear envelope has many pores formed by fusion of two membranes, and because these are the passages for the movement of RNA and protein between nucleus and cytoplasm. Next is nuclear matrix, which is nucleoplasm. It contains nucleolus and chromatin. If you see chromatin, it is a network of nucleoprotein fiber which contains DNA and basic protein histone, and it contains histone. And during cell division, this chromatin condenses and forms chromosome. Nucleolus, one or more non-membranous spherical body. Nucleolus non-membranous spherical body है, structure नहीं, spherical body है. This is important mark. And its content is continuous with nucleoplasm, and it acts as a membrane, and it is site for active ribosomal RNA synthesis. Next is chromosome. A human cell has 2 mm long set of DNA, and its 46 chromosomes a human cell has. Every chromosome, if you see the structure. Every chromosome has 2 centromers. One centromer, 2 chromatin. These are chromatin, this is centromer, and there are disc-like structure here, which we call as kinetochores. And this helps in attachment to spindle fiber. And above this chromatin, there is like this structure which we call satellite. Now see, every chromosome has a primary constituent centromer. On the side of centromer, disc-shaped structure called kinetochores. And based on the position of centromer, chromosomes are of 4 types. Metacentric chromosomes. Metacentric means where the centromer is in center. See here. In submetacentric, the centromer is slightly away from the middle of the chromosome. In acrocentric, it is close to its end, forming very short and one very long arm. Fourth is telocentric, which is at the end, not close to end, at the end. You can see here, this is metacentric, subcentric, acrocentric and telocentric. Short arm we say P arm and long arm Q arm. Next is microbodies. These microbodies, these are membrane-bound minute vesicles which contain various enzymes. Various enzymes like first is peroxisome, second is glycoxisome, third we can say cellulosome. Let's see the comparison between plant, prokaryotic and eukaryotic cell. Prokaryotic cells are generally smaller as compared to eukaryotic cells. In prokaryotic, genetic material is in form of nucleoid, and in eukaryotic, it is in the form of nucleus. In prokaryotic cell, nuclear membrane absent. In eukaryotic, it is present. Membrane-bound organelles are absent in prokaryotic cells, whereas in eukaryotic cells, it is present. Means membrane-bound organelles are present. In prokaryotic, we can say there is circular DNA, whereas in eukaryotic, there is linear DNA. If we talk about the type of ribosome, in prokaryotic, ribosome is of 70S type, and in eukaryotic, it is 80S type, which in here we have completed our first chapter of 11th NCERT. And if you like my video, do share, subscribe and hit the bell icon. Thank you for watching.